G'day everyone. Today's Chem Help video is about proteins. The aim of the video is to give you some background about the important things you need to know in exam about proteins and how to explain some of the key points. So let's get started, hey? Alright, first of all we need to start off with this, the amino acid. Amino acids are naturally occurring in the human body. There are 20 of them. All 20 are the same except for this R group. This is the thing that differs. Now you don't need to remember them all, they're all listed on your data sheet, so don't spend hours having a look at trying to remember the different ones like glycine and lysine, etc, etc. Use your data booklet for that. What's interesting about an amino acid is that there is an amino group and a carboxylic acid group. Now you might say, well, why is that important? Well, in the alpha amino acid, which is where the NH2 is adjacent to the carboxylic acid, we have a molecule where we have an acidic part. Now remember, an acidic part means something that can donate a proton. And we have a basic part, which is something that can accept a proton. So within the one molecule, we've got a proton donor and a proton acceptor. Now that makes them quite unique in terms of a chemical. Now that comes into play later on. So there's the amino group, there's the carboxylic acid group. Now what happens is that the proton can be donated by the acidic group and move to the amino group and it'll accept that proton. And what that produces is this thing, which is really important and is unique. It's called a Zwitter ion. A Zwitter ion overall is neutral, but it has a charged portion and a positively charged portion and a negatively charged portion, and that makes it quite unique. This is the form that it's going to exist in, especially in alpha amino acid where they're adjacent because it's quick move across. All right, so when you're drawing an amino acid, this is the preferred natural state. The previous state I showed you, the neutral molecule, actually doesn't occur that often. When we draw them for proteins, so we're going to specifically talk about proteins now, it's a good idea to actually draw them like this. It's a slight change from what I had before. What I've done is I've had the carboxylic acid on one group and the amino over here, or the carboxylate ion and the amine over there. Okay, so what we've got is in a straight line with the R pointing up and the hydrogen pointing down. Now, if we add two Zwitter ions together, or two amino acids together, we get a dipeptide, 2 units. Now, what's really important here is that when we added the two together, we lost the O from here, and we lost 2 hydrogens from here. And what we create is this special bond here. This is called a peptide linkage. And this is really, really important bond within the protein structure. This is not a protein, by the way. Proteins are very, very long. Almost hundreds of amino acid groups going on and on and on and on. We also get water. Why do we get water? Well, water is the residue that comes from the O and the H2 coming off here. This is a dipeptide. Notice I've still shown either ends as in the Zwitter ion form. So that's a dipeptide. Like we make a nylon, we would make a protein or a dipeptide turn into a tripeptide. So here we've got 1, 2, 3 amino acids in a line, okay? Making our tripeptide, and again, every time we add an amino acid to our peptide, we lose some water. So that's why we describe that. Now, a protein would look like this. The difference is that a protein can have NH2 plus and O minus on there, but this is a portion of a protein. It's like they've taken a snapshot of it. So before I had the peptide, all right? So the peptide is a molecule itself. Protein, now let's say a portion of a protein looks like this. Now we can't show the whole protein because it's hundreds of amino acid residues long. What's important to remember when we're looking at a protein is that there's no N there like we would have in a polymer. Why is that? Well, in a polymer the N repeats, but an amino acid is a random allocation of the 20 amino acids. So they're changing quite frequently in the human body. When you're making something, the ribosomes, as they're putting together the amino acids, they put them together in a different sequence. So to say N would mean it'd be repeating itself. We don't have that, all right? So we don't put the N, but we're showing the brackets to show that it's a portion and it continues on. Okay, so a couple of important things. That, remember, the CN, that's the peptide linkage. Now we come to our first level of structure here, and that's this. This is the primary structure. There are 4 levels of structure. In year 12, you only need to know about 3 of them. Primary, secondary, and tertiary. Quaternary, it's not on the syllabus. All right, so the primary structure is a covalent chain. Now remember, covalent bonding is very, very strong. So to break this requires immense amount of temperature. So when you hear about proteins denaturing or cooking eggs and stuff like that, you're not going to affect the primary structure, which is the covalent bonds, because that takes a lot of energy to break those, remember. Think about diamond, that's covalent bonds, lots of energy. This is a carbon-carbon bond, same thing, all right? So it's very hard to get rid of the primary structure. It's the sequence of amino acids. So that's the first level of structure. The second level of structure I'll highlight like this. Okay, so here's our amino acid, once again. We've got a hydrogen there and an R there. That's just to show you that you can have different groups there. All right, now if I have another protein or peptide, so we're showing proteins here, and I've just drawn it, it's the same structure, just drawn it slightly differently, lying nearby. All right, so here's one chain, here's another chain, or it could be it's one chain coiling on itself. You notice we've got NH and CO. Okay, now NH, this is a hydrogen bond donor site. CO, now CO doesn't create hydrogen bonds, but the two pairs of lone electrons that would be here makes it a hydrogen bond acceptor site. Hydrogen bond donor site, hydrogen bond acceptor site. Now when the two protein chains lay near each other in this fashion, all right, what we can get is we can get interactions. Hydrogen bond donor to hydrogen bond acceptor. Hydrogen bond donor to hydrogen bond acceptor. Hydrogen bond donor to hydrogen bond acceptor, and it goes on, all right? So this is our hydrogen bonding. Now this interaction between the NH, hydrogen bond donor, and the CO, hydrogen bond acceptor, keep saying that, it's really important. These hydrogen bonds are responsible for the next level of structure, which is your secondary structure. Secondary structure takes two main forms. We've got an alpha helix, which is essentially where the chain coils on itself, and you get approximately, no, not exactly, 4 turns. So 4 amino acids in every turn, it's 3 point something. That's the alpha helix. Or you can get the secondary structure as a beta-pleated sheet. So here is a separate portion of a protein, and here's another portion of a protein. Now the R groups are pointing out in this case, or along the plane, but we've got these hydrogen bonds here, and it kinks up in a pleated sheet. So a helix or a pleated sheet. Now the R groups have some influence about which one it takes, but there's no general rule for it. Some R groups, beta sheet, some R groups, alpha helix. It all depends on the portion of the protein itself. But that's the secondary structure. Now when you're denaturing a protein, this is one of the things that can be disrupted by temperature. Okay, so temperature increases the average kinetic energy of the particles, increases the vibration of those particles, so will weaken hydrogen bonding, so the coil, helix, or the sheet could be disrupted by temperature. Okay, the tertiary structure of proteins is the next level. This is the most important level of organisation, as this is the 3D shape. And that's what happens when denaturing occurs, as well as the secondary bonding being disrupted. The tertiary bonding is being disrupted. So the most important level is this. The overall tertiary shape may be long and narrow, so for fibrous proteins, or globular for things like enzymes. There are 5 types of interactions determined in the tertiary structure of interaction, all right? And these are due to the R groups. So primary has nothing to do with R groups. Secondary has nothing to do with R groups. Primary is covalent. Secondary is hydrogen bonding between the amino acids. Now, tertiary is the R group, so this is where these are important. Okay, so the first one of those forces is dispersion forces. So of the amino acids, of the 20 that we have, some, like phenylalanine and valine, have groups on them that aren't polar, aren't ionic, don't have anything that can covalently bond, but they've got large molecules like this benzene ring and this methyl group, or this chain here. Now, there's nothing polar about those, but they've got lots of electrons. So they have strong dispersive forces. So when these portions come close together with each other, we have dispersive forces. And remember, dispersive forces are there all the time. And it's really important that you mention that in things when you give descriptions, because we've got this cumulative nature of bonding. We can have dipole-dipole forces. So as we know, hydroxides can hydrogen bond, but they're also very polar. So you can have polar bonding between serine and threonine or other groups. as I said, hydrogen bonding. So we've got an OH, we'll have a hydrogen bond donor. We've got a hydrogen bond acceptor here, hydrogen bond donor there. So serine and asparagine, they can hydrogen bond. So same things you talk about when you talk about any type of interactions, dispersive, dipole-dipole, hydrogen bonding, they occur to the R groups as well. Now we get ionic interaction as well, and that's where you've got parts of molecules that are ionizable. Alright, so now this is aspartic acid here. Alright, CO, it's not an ion at this stage, but you remember that weak acids ionize partially in solution, especially if there's a base nearby, but they do it with water anyway. So this COOH group will turn into a carboxylate ion. That means it can ionically interact with something else, especially if it's near something like lysine, which has got an NH2 group on it. Now remember, NH2 group is basic. It can accept a proton. So if these two chains came close to each other, the hydrogen could leave the acid, go to the base, form ionic interactions, and come together. And that's why these ones are really important when pH is being considered. So if you've got a amino acid with R group interactions holding it together in a certain structure, and you add acid to it, you might denature the enzyme. So enzymes don't work well in acidic environments. And you say, well, why is that? Well, what happens is that the added acid will force the weak acid to reform, because we know weak acids are the predominant species in solutions when it comes to these things. So that means that would form, which means the ionic interaction would not be there anymore, and therefore the tertiary structure would be affected and the enzyme will not function. So ionic interaction is really dependent on pH. So be careful of that one when you're doing what is the effect of pH. Covalent bonding is the last type of tertiary bonding. All right. Now what that happens is when you form covalent bonds between chains, and it happens a lot with something like cysteine. It's got a sulfur group in there. Now what happens is you can form disulfide links, which are cross links, which are covalent cross links, which are incredibly strong. Remember, covalent bonds are the strongest of all the bonds, and that means that they can really be held together well. And that means that it can't be affected by pH, can't be affected by temperature that well, because temperature requires a lot of thermal energy to break a covalent bond. So this means great stability between amino acid chains. And that essentially is the last one. Quaternary structure is a lot more complex and not covered in the Year 12 syllabus, so you don't have to worry about that. Hopefully that's helped you out. And I'd like to thank you for watching this one. Give a shout out to some of my students who are finishing up in the next couple of weeks. So good luck, guys, and we'll talk to you next time. See you later.